Welcome everyone to this spotlight on the first unit on this PG Certificate in Clinical Bioinformatics. My name is Professor Ang Davies and I'm Professor of Clinical Bioinformatics and Healthcare Science Education, and I'm the unit lead along with Andrew Devereaux for this introductory unit in Clinical Bioinformatics. Well, first of all, we'll introduce the unit aims, the learning objectives, and a little bit around the key course content. We'll talk about genetics and genomics, of course, and we'll give you an introduction to molecular biology and the scope of genomics. We'll also begin to introduce sequencing, so the types of sequencing, applications and limitations of different sequencing approaches. And then we'll really delve into the fundamentals of bioinformatics. And this includes the sort of introduction to the history and the scope of bioinformatics, including its clinical application, and also a little bit around ethics, standards and governance. So what are the aims of this particular unit? Well, fundamentally, we want to provide you with a background knowledge of human genomics as applied to a clinical setting. We'll talk about transforming patient care using technologies like next generation sequencing or NGS. We'll delve into some of the basic concepts around next generation sequencing and how we analyze genomic data. And we'll also begin to introduce some of those key bioinformatics tools, databases and methodologies that we use to really interrogate and understand clinical genomic data. To start off within the unit, we'll cover some key concepts around genetics and genomics. So we'll introduce you to the genomics landscape with a particular focus on the UK, but we'll also ask you to think about the genomics landscape in your own countries. We'll take a look at nucleic acid structure and function, and we'll think about that in the context of the central dogma and how we go from DNA to RNA to proteins. We'll also look at things like non-coding regulatory sequences and the role that they have within genomics. And then we'll begin to look at Mendelian inheritance and the role of Mendelian inheritance in the context of rare inherited diseases. Then we'll look at how variation is integrated within the genome and its role and relevance to health and disease. We'll also then begin to introduce genomic technology and the role of the genome in the development and treatment of disease. So one of the key ways genomics has changed is through the application of next generation sequencing technologies over the last sort of decade or so. So we'll really in this unit begin to introduce these sequencing technologies. We'll look at traditional Sanger sequencing versus short read next generation sequencing versus the long read technologies that are now beginning to come in like nanopore and PacBio and things like that. And we'll look at the advantages and disadvantages of kind of all approaches. Moving on from the sequencing, we'll also then begin to look at the analysis, the annotation and the interpretation of that sequencing data that comes off of the machines and how we use that within the clinical setting. Within the context of next generation sequencing, we'll also take a look at the different kinds of ways we can use this in the clinic, from gene panels through to looking at focused exomes, through to now looking at the whole genome. So we'll begin to look at some real fundamentals of bioinformatics within this unit, and later on in the course we'll really start to build this into some of the pipelines and analysis of next generation sequencing data. There are really sort of three main aspects we look at within the course. We look at the databases that we might cross-reference to analyse genomic data and interpret variation in the genome. We'll look at other kinds of tools you can use to do that. And we'll look at ways to visualise genomic data. So in particular, we'll look at various browsers, including Ensemble and UCSC, to be able to visualise genomic data. And we'll bring in some other tools as well. Other tools we might look at are Nomad. So this is where we might look at particular frequencies of different alleles, and also OMIM, where we can find out much more detailed information about the phenotype of particular genetic disorders, and other bioinformatics tools that we might use to look up sequence information, to generate alignments, such as BLAST, Clustal Omega, and other tools like that. And then the real key part of this course that we'll focus on is applying those tools and databases to real clinical case studies. So we'll assign you to a particular case study where you'll study a variant that's been sequenced within the clinic in our own laboratory in Manchester, and we'll ask you to really interrogate and interpret that variant in the context of that particular detail of that case study. So the key difference to this course in comparison to many bioinformatics courses that you might find is it's focused around the clinical application of bioinformatics, which is kind of a discipline in its own right. And because of that, we're focused our learning around real clinical case studies. So these are case studies that you might come across and work on within a clinical setting. So we'll be asking you to select and apply appropriate bioinformatics tools and resources to typical diagnostic laboratory cases that you would come across in the clinic. And then importantly, we'll teach you and provide you with the materials that will enable you to interpret the evidence from various bioinformatic tools, resources, the literature, and other sources to really interrogate that evidence, and then you'll be asked to present that within your final assessments. So a thread that flows throughout the course is really to remember to think about the ethics, the clinical standards, and the governance surrounding clinical genomics. And in particular, as we begin to develop more and more clinical bioinformatic pipelines to analyze data, we have to come back and remember ethics and standards and governance. So in particular, we'll apply certain international clinical reporting guidelines. In particular, we focus on ones that are provided by the American College of Medical Genetics and Genomics, and we'll use those and apply those to our case studies as we're working through them. So all the time we're thinking about these clinical reporting guidelines and what clinical laboratories actually use in the real setting. And we'll use those guidelines as we work through those case studies. So we'll appraise ethical issues related to the mainstreaming of genomic medicine. So this means as we use it more and more across the hospital setting, as we apply techniques like whole genome sequencing, we have issues around finding more and more secondary findings. So these are issues not related to the patient's phenotype, but that might crop up and actually have clinical implications. So we'll think about how we deal with those kind of issues. And we'll also think about issues around reanalysis of data. All the time we're finding out more and more information about clinical variants. There's more information coming through in the literature, in the databases, the tools. So how do we deal with that amount of data, and how do we cope with reanalysis within the clinical setting? So how is clinical bioinformatics developing within the genomics landscape? Well, first of all, certainly in the UK, we're now seeing mainstreaming of this service and the application of whole genome sequencing in the clinic. This is a massive change over the last couple of years. It was propelled by the 100,000 Genomes Project. And what that means for bioinformaticians is that in general, the pipelines are changing. There's far more data that's coming through, and there's lots more variants that need to be analysed. So all of that needs thinking about. We're moving to things like cloud solutions to deal with some of these changes. And all the time, new pipelines are coming in. They need to be validated. They need to be updated. So people's skills constantly need updating. In addition to that, we'll be analysing that genome for new types of variation. So there's a type of variation called copy number variation. It's quite often found to be linked to genetic disorders. And more we're seeing much more of a pull to develop that kind of application into the genomics pipelines that our bioinformaticians are beginning to work on and to develop. And also as the profession develops now, we're seeing many more applications and developments of new clinical guidelines and quality standards that really govern this area of clinical bioinformatics in much more detail. Okay, so how does this course help with career opportunities? Well, for sure, with all of this mainstreaming of genomics, the growing pipeline that's being applied clinically in terms of whole genome sequencing, there's really definitely a growing requirement for clinical bioinformaticians that are needed to analyse all of this genomic sequence data. In addition to that very clinical setting, there's also sort of much more translational research bioinformatics roles, which span research across to application into clinical environments. And so these are also kinds of roles that we see some of our students going into. We have some laboratory scientists that are quite often interested in the course, sometimes exploring a change of focus or roles, or actually just really wanting to understand much more detail around the bioinformatics that relates to some of the upstream work that they're involved in. And then we also have clinicians, sometimes some of the end users and receivers of some of this genomic data. they sometimes want to do the course because they actually want to understand much more about their patient's genomic data and how that data has been processed before it's passed over to them. One of the other real advantages of this course is that we apply all of the assessment and learning around real clinical case studies, and we ask you to work together in groups. We ask you to present your work in a recorded way to other peers and students, and we also ask you regularly to contribute to discussion boards. And all of these ways of applying and synthesizing this knowledge are developing key transferable skills that will help you to present some of this understanding, for example, for people working in multidisciplinary teams in a clinical setting, for those wanting to use those transferable skills to potentially apply for slightly different roles. It's all a really good background if you're thinking about any of those slight changes of direction or application for different roles. It's really great that throughout the course we have lots of discussions through tutorials and also discussion boards where we can contribute to the learning and have a dialogue with our students. So this is a particular student that's I've worked with over the last year or so called Arjun Ryatt. He's currently a technologist, a genetic technologist, working in one of the genetic laboratories in the UK. And he's really developed a strong passion throughout his learning and application of various tools throughout the course that's really inspired him to want to apply for one of the clinical bioinformatics roles, training roles that are available within the scientist training program in England. So the scientist training program is a program that Manchester's been involved in now since around 2013. We deliver the master's program for this course, and Arjun really is passionate about having a clinical bioinformatics role in the future in the NHS. So the course has actually developed his knowledge and skills, and he now feels confident enough to be able to apply to that program. And hopefully in the future become a bioinformatician in the NHS, which is fantastic.
The thing I enjoyed the most was meeting the diverse array of students that we had taking up this course, and it was interesting to see how different people from different backgrounds were able to look at the same assessment that I was looking at and have a totally different perspective. I have to say, throughout my course I was also highly motivated by different students. They constantly supported me. It was because we were given group assessments and assignments that we kind of relied on each other, and by relying on each other we brought the best out of each other.